visualizations. And I think we will see AI moving in because lots has happened this year. We will see different, more live feeds of data come in. And I think we will see some most stunning and beautiful visualizations and stories that are told about these visualizations, and maybe multiple stories because each stakeholder might need a slightly different story. And this is beautifully leading over to the next presentation by David Chavalarias, who's joining us from Paris. And he has been contributing to the exhibit, also served as a host for the exhibit in Paris for many years. And he does beautiful, beautiful storytelling. And it's my great pleasure to have him here. David is the director of the Complex Systems Institute of Paris, Île-de-France, and he's going to present his work on storytelling. So if you can stay, please do. We have the call in the chat, and I will also put a link to David's work into the chat. So David, if you want to take it away from here, and again, thank you to all of you presenting here at the debut.
Yes, thank you. Thank you very much. I'm very happy to participate to the exciting 24-hour rally. It's very nice. So I will present very short stories about several microscopes that we are building at CNRS. So I will share my screen, hold desktop. Okay, perfect. Okay, so I built few short stories. I think that you know some visualization techniques, so I will not give some details sometimes, but if you need, don't hesitate to interrupt me. I can give more details, and all what I will show you has been made in collaboration with several people here at ESC, Alexandre Lanoé, Basia Panahi, Quentin Lobé, Victor Chauvel, among others. So the idea here is to use Macroscope a little as we use Microscope. So we are using Macroscope to look at the complex systems that inhabit us, and now we are building Macroscope to look at the collectives that we form. And what we are trying to do is to build, and we also see this kind of feature in other presentations. So to give the users the ability to go from low levels, so the micro level, to higher level of the structure of the data sets that he or she is exploring, and going through the mesostructure. So I will show you a few examples of that and different kind of application. So the first one I want to talk about is about social microscopes that I'm developing with Basia Panahi, who is building all infrastructure to collect social data. And I will give you an example on a recent case study that we did on climate change. So here's the idea was to observe climate change debates, both at the social levels and the semantic levels. And I will give you just a sense of what we discovered. So to start with a small video, I hope this goes through the Zoom well. This is, for example, what we observed where we get all the Twitter data about the climate change debates. Here, every dot is a Twitter account, and the links between the Twitter accounts are just retweet between people. And the pulse is 1 week. So every week we can grow again these networks. This is worldwide. You have a United States, France, etc., and you clearly see that is emerging two big blocks, two big groups or big clusters. On the top, you have the people that are pro-climate, and on the bottom, you have the denialists. And when you have this kind of structure and you observe this over several months, you can ask many, many questions about that. So, for example, of course, you have the structure, and inside the structure, you have different sub-communities. So again, every dot here is a Twitter account. You have different shade of colors, which are different communities, and you see that you have geographical communities, but also communities related to activism, like for example around Greta Thunberg, etc. And you still see very distinct big cluster, which is denialist and pro-climate. So one question when you can observe these kind of social systems at these scales is what is circulating inside these social systems and how the different communities organize themselves to sustain say, shapes and structures, the number of the members. So I will very quickly give you some results. We have a big report that analyze all this. But for example, there was some conspirationist narratives that has appeared in July 22, which is here you have an example of this tweet, which is just saying that now that COVID is over, the next fear factors for government will be climate. And what we saw is that this narrative that was pushed by some boats at a very precise date here in July 2022. We just pop up like two dozen thousand of robots that were just relaying this kind of narratives. So we have a burst of accounts that were relaying this, and we could analyze this account and demonstrate that all this account has an anomalies in the creation dates, which means that they are boats. And after that, we can try to see in this kind of maps where the boats were activated, have been activated. And if I highlight in this map the place where the boat have been activated, pushing these conspirationist narratives, I have this map here. So here I have just highlighted in red the boats that have highlighted this relays these narratives. If I isolate them, I have these things. So here you have at the center the ones that were really well networked in these climate skeptics communities, and around you have unconnected robots or accounts that are just coming to relay this. And from that, they push the narrative at scale, so almost 50K reaction in less than 2 days to try to influence the global networks and the algorithm, etc. So this is the kind of things that you can observe. And in general, what we built is we try to give context to people, citizens, journalists, decision makers about where a particular piece of information is circulating and coming from. So for example, with the same principle of mapping social landscapes, we had these journalist platforms during the French elections. And when a journalist came, they have this kind of thing. So I will make you a demo here. So for example, this is French political landscapes. Here, I will, when you zoom here, every dot here is a Twitter account. And for those who know this, here is the names of the main political personalities. And for example, we can ask who also relays these ideas that there was a link between the COVID and climate with this case conspirationist. So people can come here, ask something and say, okay, show me where are this guy. And you see here that this narrative is coming from this area, which is the far right in France. And so the idea is that you can ask anything like that to see where are some narratives coming from and how it spreads through your landscapes. So this is, sorry, this is not. This is one thing. The other thing is that you can look at what they are saying or what is the different discussion about climate change. And for this here, we built another macroscope, actually, which is older than that. I will just give you a few demo about that. So here, this is this platform here. So here, what we did is that we have mined about 30,000 papers, scientific papers that appear here to generate this representation where here every node is a concept that has been extracted from the paper, and the link are some kind of semantic similarities between the papers. So I guess you have seen already this map, so I don't give much details. It's more or less a co-occurrence maps. The semantic distance is a little more complicated, but so you have the semantic graphs with clusters. And one question that we ask is, so this is a map made from the data from scientific data. And on the parallel, we have this Twitter data. So we did, we look at what is the difference between the attention for the topics from the science and from the Twitter. So if I change the coloring of this map here like that, now the color of the nodes is here. This is the legends. And this means that when a node is in green, this means that the attention in terms of the percentage of documents in the Twitter database, in the scientific database that are talking about these terms, the attention is balanced. The more you go blue, the more it is talk about the Twitter landscapes, so Twitters and all the reference that you have in Twitters, and the more it is red, the more it is only talked or discussed by scientists. So from this map, I forgot to comment this map, but very quickly here you have two branches. One is the branch about the impact of climate change on societies. So you have and the impact of societies on climate change. So here you have everything about emissions and greenhouse gas and tax. Here you have everything about the impact, directly impact on the people and the urban areas. And when you are talking about urban areas, you have the impact on crops, impact on water. And the other branch is the ecosystemic branch with the ocean here, the ecosystem here, and also the soil ecosystem here. So again, if you change the color here and you have the attention of the people, what you see immediately is that people are well aware about climate change and it's linked with greenhouse gas, for example, or impact of heat waves. But when it comes to the ecosystemic branch here, then it is less clear to what extent people are aware of things, and especially in this cluster here, which is about the soils. So what you see here is the blind spots of the public opinions that is not very aware that you will have impact of climate change on the organic matters of the soils and the soil properties. And actually, this is very important because soil microorganism is about 20% of the CO2 emissions. And also, when you have this, then you can also try to play this attention. So here, week after week, the colors are changing according to how the public attention is modulating and circulating in this kind of landscapes. And you can see that here, this area is very stable. So the climate impacts on societies. And here you have some times where it's getting more bluish because you have, for example, the forums on the sea and the oceans. So we are talking about that in the media and after it stops. So this was using this macroscope to spot the blind spot on climate change, which here was the question of the soils. Now, I will talk to you about how we can do this kind of map in a very agile way with this software, which is Gargotex, which is a web service and entirely built on APIs. So it is in the same spirit of what has been shown just a few minutes ago, to be able to have different modules and connect. And I will show you some example of stories that you can tell with these kind of platforms. So it is built by Alexandre Delanoë, myself, and with many contributors. And so one of the examples that we are developing this, one of the case studies is trying to reconnect science with public policy. So give easy access to, for example, political figures when they have to take a decision, for example, for the authorization of an herbicide like paraquat, or to think about the kind of products that you have in air dye. So how they can connect with actual scientific materials to make themselves to make an idea. So I will show you two quick examples. So the first one is about paraquat. So paraquat is a very widely used herbicides, including in the United States. And you have about 9,000 papers in PubMed. And the question you can ask is, what scientists are telling about this? Because there is regularly the question about, should we forbid this substance or not? So what we do is that Sorry. Yeah. Okay. I will. So now I will show you. I show you the inside of the Gaia context. So this is a web platforms that where you can import documents. So here, this is a document about Power Query that we have imported from PubMed, and the importation is really easy. You have a direct API connection. You make your queries, and it comes inside this software Gaia context. So you have the documents. You can read the documents inside the platform. So for example, here I just read one text, and you have the text can be annotated. So you can ask, for example, you can say lifespan is an interesting concept. I want to have this, and superoxide generators, etc., etc. So you can read and annotate. When you have read and annotate things, then you can generate the maps. So here I will show you. You just click here, will generate the map specialized. So this is generated on the fly, and this will take into account the last annotation. So I will not wait for the specialization. I go directly to the specialization, but you will have the same things. And here, so this is some kind of networks representation that you should all know. But you can ask now something about this. So you have the different areas about paraquat. So you see that we are talking a lot about toxicities. We are talking about lungs here. Here you have talking about the impact on the cells, and especially the oxidative stress effects. And here is you have the action about how you can cure paraquat and what is the mechanism on rats. This is analysis on a specific in labs on specifically on animals. And here you see that there is some talk about the Parkinson. So from this map, then you can query, and you can directly from the map ask for more. So here I have selected the Parkinson. So I see that when you are talking about paraquat, there is a link with the Parkinson, and actually it's one of the arguments to forbid paraquat. And you can directly have access to the original paper here that deals about this topic. So you can directly come back to the source, read the source again, annotate again, and make the map. So when you have this, the question is that I have the question of Parkinson here, but to what extent this topic is new, how it is, has this been resolved? This is some emerging concerns about paraquat. So here I will show you another visualization, which is in Gaia context, which is a phylogeny reconstruction. So here, what we have is that I have small dots here, which are supposed to correspond more or less, but at a finer granularity, to the color here. So here in each colors, every colors here is a topic that way of speaking about paraquat. And here, this is all these terms of the same colors are gathered inside a circle like that. So here you are talking about a neurodegenerative disorder, Parkinson, etc. And here you are in 2006. What we do is that for every year, we do this mapping process. We detect the colors, and we make all the bubbles. And you see that from one year to the other, then you see that this cluster here has been some ramification. So you see here, we are more or less the same topic here, but here it is linked to herbicides, also Parkinson, etc. And we make this vertical links, which materialize the relation, semantic relation between two topics at two different time steps. So here we are in 2006, here in 2007, here 2008. So if I zoom out, I will have all the topics that have been detected for all the periods and their relation, intertemporal relation. And this gives us the phylogeny, what we call the phylogeny of paraquat, And you see here that this topic is unfolding, and also there is more and more discussion about different things. And here you are like 50 years about paraquat and paraquat delays. And if we look at this Parkinson things, so I click here on Parkinson here, I will highlight all the subtopics where we are talking about Parkinson, and you see that it is since 2005 that we have started to think about the link between paraquat and Parkinson. Here you see that it is linked to neurodegenerative, to exposure, neurodegenerative, impact on dopaminergic neurons, oxidative stress. So this is all the terms that have been related to Parkinson. And you see that this is actually an emerging trend. And there is more and more topics talking about that with different variation, different topics. So this means that you have the question of the pathway, different analysis. So this point to the fact that there is a really concern about that. The other quick example I show you is about a substance that is used in color dyes. So I will go here. So same thing here. This was just a question I asked a colleague that told me that there might be some problem with this component here, which is used in color dye. For example, in this product that is first in the USA. And so I say, okay, let's import this from PubMed into this guide context. So we have about 1,200 elements, and you generate the graph. And what you will learn from the graph, so again, I will go directly to the one which is parcellized, is that if you look at how this substance is used for hair, so you see that it is heavily used in hair dye. And if you look at what are the closest terms associated with hair dye, all the expression about hair dyes in this corpora, so you see that you have dermatitis, allergic contact, disease, you even have death. So if you click at death, then you see that it is linked to cancer. And actually, this PPD is short terms for this substance. So you see that there is lots of studies about this substance causing some cancer in the long run. And actually, this substance has been forbidden everywhere except in hair dye. And from that, then you can look at the direct scientific records here and make your own idea about is there some serious issue to forbid or not this thing. So here the idea is to bring this kind of macroscope to decision makers so that they can easily connect with science and public policy and make their own ideas and not be so shy about going directly to the original paper. So I will finish with, if I have time, it's okay. I've very quick example to finish. So the idea is that what I show you only use text mining, direct text mining. We don't use any other metadata. So we can apply this kind of approach to any kind of corpora that have text string in it. So I will show you just a few examples where we have nice stories to tell. The first one is clinical trials. So you have registries of clinical trials that is published by the World Health Organization. And during the COVID-19 crisis, we have monitored the evolution of the clinical trials. Clinical trials are very interesting because first, when you have to make some clinical trials to start developing a vaccine, for example, you have to, before everything, post the description of your trials on these archives, and you have this kind of things which explain you what you intend to do and what you intend to test. And this is usually 1, 2, sometimes 3 or 4 years before the actual publication. So when you look at clinical trials, you have this. You are still here, just because I don't see. Yeah, okay. So I did several maps about coronavirus. For example, this is a map about what we were talking about coronavirus very early in March 22. I won't comment this map, but just to highlight that it is really easy to make new maps and ask questions. I go to the clinical trial directly. So when you have a clinical trials, in the clinical trials, what you have is some description about few substances that you are testing on human. So for example, here you have a clinical trials that has been run in March. Here you see 5 March, it's a week of the 5 March, it is 2020. And again, we detect all this. So here, the clusters are very smaller because typically in a clinical trials, you are testing 1, 2, or maybe 3 substances at once, but rarely more. But still, you can do this intertemporal matching. And when you do this on clinical trials, you get this kind of representation here, which is here. These are all the branches of clinical trial testing until June 1st, 2021. And what is interesting here is that you see the different paths of research about testing these COVID vaccines. So, for example, in this branch, it is all the vaccines that were testing, all the pre-existing vaccine that has been tested to see if they, by chance, they would work on COVID. So you have BCG vaccine, you have measles vaccine here, but as you see, so times goes from top to bottom. So as you see with time, this branch fades out because actually no existing vaccine was working. And in parallel, you have new branches here that are appearing that actually end up to be the promising branches. And this is in June, and you have one branch here about non-replicating viral vectors. You have another one here about protein subunit, and this is RNA-based vaccines with, for example, the Pfizer one. And you see that there are no connection between these branches because at that time, every labs were testing its own technologies. But if you go more in time, so now we are in July, in September. So in September, you see that these branches are beginning to connecting with each other. And what happened is that you have this ETI label, which is for heterogeneous clinical trials, where you are testing different products. So if you look at the ETI, I click here on the ETI to highlight here. Or I will do it like that. I don't know why it's not working. Yeah. So here you see that after you develop the Pfizer, the AstraZeneca, etc., then you will try to test the combination of them, and this makes this branch to join. And so here you can really see how the science is evolving, starting from simple substance and then trying to combine different substances to see if it is more efficient. And so we can comment more on this figure, but I pass to this other example. By the way, you can ask if we could have anticipated that the pandemics could be one of the big risks for our societies, and very quickly we had has already done maybe 5 or 7 years ago cartography of all the risks from the scientific literature. And when you do this, you see that the risks are divided into 2 big groups here. At the bottom, you have all the risks that are linked to related to health. So this is from genetics, and this is from HIV, drug use, etc. So the bottom is health. The top of this map is all the risks that are related to environment and ecosystems. So this is soil ecosystems, this is climate change, and this is financial markets. So you have these two big clusters that seems apparently disconnected. But here, this map has been drawn after some resolution. Here, I only show the top 1,000 terms. But we can go more deeper and ask if we add more terms and more topics, which are less popular, let's say, can we populate this area here? If we do that, then we have this map, which is the same, actually, it's just symmetric, but it's really the same. At the bottom, you have health, at the top, you have ecosystems, but you have a new cluster here that is appearing. And actually, this new cluster, what is it? This new cluster is pathogens. So if you look at here, it's exactly about virus pigs. Actually, the coronavirus was known to be a pig illness. And what you see is that all these zoonoses and virus things that develops in between human and animals are the ones of the category of risks that are relating individual health and collective health. So it has been said, of course, not by me, but by other people before, that pandemics was one of the major risks of the societies. But here you understand why. It's because they are connecting two huge categories of risk. And last, to finish, I am just to show you an application of these two very different kind of data. You can look at, for example, Twitter data. So, for example, there was this very both funny and also, I cannot say in English, but quite miserable.
David, if you can wrap up in 1 minute, that would be wonderful. We all have been very much enjoying your presentations, and I think there will be many synergies with some of the previous speakers. But we have another panel starting, and Dan will take it.
Okay. So I will pass this just to say tell you that you can apply the same thing on Twitters, and if you have this on Twitter, then you have also Twitter database like that. So this is the It's the Babies Polylux that was launched to attack the Democrats from some think tank. And so now, so if you want to know more about this dynamic reconstruction, this is about the phylogeny. You have the reference here. And just, I think that with this kind of approach and also all the macroscopes that we have seen during these 24 hours, we are at the point to achieving D'Alembert's dreams, which was one of the founders of the French Encyclopedia, that wanted to make like a genealogical or encyclopedic tree to gather all the knowledge in the same place. And I think that now we have the technology to do that and to do that collaboratively. Okay, thank you very much. Also, thank all my collaborators that helped me on all this wonderful stuff. And thank you for your invitation, Katy, and the team.
David, great presentation. Thanks so much.